we are looking at is a chart whereby you see that the demand for the various these recombinant therapeutic proteins is to the order of 40% in the US, and similarly it is of the order of 7% in China. In European Union, there is a demand of about 25% for various recombinant therapeutics, which includes insulin or recombinant proteins or vaccines or monoclonal antibodies. And in all other countries, it is of the order of 28%. Now, what is extremely important here to see is that if you look at, for example, what are the expression hosts which are being used for making these recombinants, is that the green part you will see is the maximum one, which actually refers to, as you see, mammalian cells. Okay. And so what it means is that when you are trying to look at recombinant therapeutics, there are still a large number of these proteins which are going to be expressed in the mammalian cells. All right. And I'll give you an explanation for that in a while. And the light blue that you see here, for example, is all other systems, which could be, for example, Saccharomyces cerevisiae, or which could be, let's say, insect cell line, or it could be something else. And in this particular chart, what you will see is that basically E. coli, after the mammalian cells, the E. coli as well as the yeast Pichia pastoris enjoys a large amount of share in the total production of the recombinant therapeutics. So in other words, 53% of the current recombinant therapeutics are being produced either, for instance, in large number in both E. coli as well as in Pichia pastoris. Okay, so that means that there is a lot of thrust on using microbial systems, particularly E. coli as well as Pichia pastoris, not Saccharomyces, which is the common baker's yeast for production of the recombinant therapeutics. Okay, so so that is the reason why we say that it is always better to use very, very small microbes like bacterium or like use Pichia pastoris. Now, I had also told you that a large number of proteins are expressed in these mammalian cells. And the reason is that currently there is a great demand for monoclonal antibodies, and which can be used as therapeutics, and they are in a position to bind to certain pathogenic proteins inside the body. These pathogenic proteins are secreted by viruses or something, and they are the disease causing agents. So if you have these antibodies which are going to be used as therapeutics and which are going to be targeted particularly those infection causing proteins, then you directly have a way of attacking that particular target. Okay, so it is not like a drug which is going to be very, very non-specific in action. Many of them, like antibiotics, can act against a whole range of these bacteria, and they are not really specific to one particular type of a bacterium. Whereas these monoclonal antibodies, which you see here, which is abbreviated as MABS, and these are produced by mammalian cells. And the reason is that there is the issue of glycosylation. So these monoclonal antibodies are basically glycosylated proteins, and if they are produced in a system other than the mammalian cells, then the chances are that, like in E. coli, they will not get glycosylated, or in E. coli, which is the orange part here, they do not get glycosylated, or they get alternatively glycosylated, which is not good enough for them to retain their biological activity. Okay, and that is the reason why you see a very large share being occupied by mammalian cells, because they are primarily used for making monoclonal antibodies. So some of the top recombinant therapeutics are listed right here, and I will just show you, and you will also be familiar with many of them, like human insulin. It has a large share of the market amongst the recombinant therapeutics, and it is used for the treatment of diabetes. Similarly, erythropoietin is used for the treatment of anemia. Human growth hormone is used for treatment of dwarfism. Similarly, interleukin-2 for renal cell carcinoma. So in this way, a large number of these recombinant therapeutics are described here, and you will see as to for what kind of a disease these are going to be applicable. Now, what is extremely important here is that, you see, when you look at all of these, they are all proteins, so they can be coded for by a gene. So when we say recombinant therapeutics, That means we have the ability to clone the gene which is coding for these particular proteins in an expression platform, which could be either an E. coli, it could be a yeast like Pichia pastoris I showed you in the last slide, or it could be a mammalian cell. Okay, and that is why it is application of recombinant DNA techniques is possible here, because in this what is involved is that you need to just clone the gene which is coding for this particular therapeutic protein into a system. Then that bacterial or a yeast system can be cultivated on a large scale. It can be cultivated in huge bioreactors, and at any given point of time you can make large quantities of this protein available. So now let's move to the next slide. Now in your textbooks, you have been given an example of genetically engineered insulin. And I'm just going to explain to you as to what different steps are involved while you try to make recombinant insulin. And the platform for expression which is being demonstrated in this slide is that of E. coli, which is a simple bacterium. Now if we examine insulin, alright, insulin is first secreted inside the human body as a proinsulin, and the structure of that is shown here. So it's a large single polypeptide chain which is beginning from here, and then it just goes up to this part which is the C terminus. Now during the maturation of insulin, this is what we call as proinsulin. So it is a precursor of insulin. During the maturation process, what happens is that this pink part of the polypeptide chain, which is also called as a C peptide, and it is made up of 31 amino acids, is going to be cleaved out, and what is left behind are these two different polypeptide chains which are called as A and B. And the way that they are held together is through these disulfide bonds. Okay, so in the process, therefore, it is not just a question of making a protein. It is also a question of processing it correctly in order to give rise to the mature insulin. And then this C peptide, as you see, which is originally in the proinsulin, doesn't appear in the final protein. So now in the when you look at, for example, how such a protein can be made through recombinant routes, what you find is that now given the existence of this proinsulin as is shown in the figure, and then cleavage of the same to generate chain A and B, which are further disulfide bonded, there are two methods which are employed in various pharma companies in order to overproduce it. So the first way is what is called is cloning this entire gene which is coding for the proinsulin, and then followed by processing. Or else in the second method, which I'm going to demonstrate in this figure, is that you could have A and the B chain, which are like synthetic DNA fragments, which are going to code for A chain and B chain. Once you produce them separately on two different plasmids in two different hosts, then these proteins could be now brought together. They could be assembled in vitro. Okay, so both the platforms are used. So in the first method, where you have, for example, proinsulin, which is being cloned. So essentially, if you are going to use E. coli as a platform, you can imagine that now you have a gene which is entire gene which is coding for this particular proinsulin is going to be cloned into a vector. This recombinant vector is now transformed into an E. coli cell, and so you have the way the recombinant vector is going to be designed is that this recombinant protein now, which is your proinsulin, is going to be secreted inside the cell. There is no leader sequence to take it outside of or into. In the case of a bacterium, it could also go into the gram-negative. If E. coli is a gram-negative bacterium, so it could go into the periplasmic space. Okay. However, in this particular case, the strategy which is followed is that you are going to overproduce this proinsulin inside the bacterial cytoplasm. Now, many a times when you overproduce such proteins in a bacterium, it doesn't remain in the solubilized form, but rather it aggregates together because the cell is not capable of handling such large amounts of protein and to fold them properly. So these proteins are now going to aggregate together into what is called as an inclusion body. Okay, so what you do is that you are quite aware that this bacterial cell in the cytoplasm has inclusion bodies. All right. So what you do is that after this protein is produced and the bacteria have also grown to high cell densities, you are going to harvest the cells, which is followed by disruption of the cell. And now you have insulin precursor. Why? Well, because we are dealing with pro-insulin production here. Okay, except that this pro-insulin is now in inclusion bodies. There are methods which are available by means of which these inclusion bodies can be separated out from the cell cytoplasm, and then they can be solubilized. Again, because inclusion bodies is nothing but these are aggregates of proteins, and which could also have some other cellular debris like RNA or DNA or some other kind of proteins with them. So these are small aggregates, and you recover these inclusion bodies and now you solubilize them by using a denaturant like guanidine hydrochloride or any other kind of a detergent. So this proinsulin is now going to be available to you in the soluble form. And but however, still it is, so you do have a proinsulin which is now unfolded. All right, so then there are several strategies which you are going to apply at this particular point, and then you get this proinsulin, but now which is in the folded form. Okay, and then through further processing, etc., which involves cleavage of the proinsulin and release of chain A and B, and disulfide bond formation also occurs here. So after processing, which is applied at this particular stage, you get what is crude insulin. Okay, and all of these things are being done in a test tube, and eventually then through application of any one of the protein purification methodologies, this is going to be purified, and then this is what is available in the market. Okay, so in this particular example, what we have seen is that you are going to overproduce proinsulin. The picture on the right shows you how the second strategy can be adopted, where you have now a plasmid which contains the synthetic gene fragment which codes for the A chain of insulin. All right, you also see a yellow segment here. What that is a part of the beta-gal gene. So in other words, what we are trying to do here is constructing a fusion protein. Okay, now this is necessary, especially in the case of E. coli, because beta-gal, the way the sequences are, amino acid sequences are there, or there is a leader sequence ahead of it. Okay, now the recombinant E. coli is tricked into believing that it is probably transcribing and translating one of its own proteins, because the cellular machinery has the ability to recognize the self proteins and the non-self proteins. Okay, so what was found over over research which has been carried out over decades is that if chain A is now synthesized as a part of a fusion protein with beta-gal, then it is going to make large amounts of this fusion protein. Then number two, we are going to develop strategies to separate out this beta-gal part of the fusion protein. So that is the kind of a strategy which is applied here. So you have a recombinant vector like you see here. So this is your chain A of insulin, which is preceded by beta-gal. All right, and then there is a leader sequence also here, as a result of which the fusion protein is going to be excreted out into the cellular medium. It doesn't stay inside the E. coli, but it comes out in the extracellular medium. Similarly, you have another plasmid vector like you see here, which codes for chain B. Okay, so obviously it is a synthetic DNA fragment, which is going to contain a nucleotide sequence, which when the triplets are decoded will translate into a sequence of amino acids in chain B. It is also constructed as a fusion protein. So you have these recombinant vectors. So this recombinant vector is now transformed into an E. coli cell. So you will see that the plasmid is right here. This is the chromosomal DNA. Similarly, this particular recombinant vector is transformed into E. coli cell, and you have the recombinant vector here, and you have the chromosomal DNA. Okay, now because of the leader sequence, the fusion protein is secreted out of the medium. All right, this is the way it is going to look like. So the yellow part is the beta-gal part, and the blue part, what you see here, is the chain A of insulin. Similarly, from the second host also, you are going to get a recombinant fusion protein. So you have the beta-gal part, and then you have the chain B part. Okay, now the amino acid sequence which is at this particular site, at the fusion site, is such that it can be cleaved now by cyanogen bromide treatment or whatever, because they have a specificity to act at methionine residues here. Okay, so there are several protease protein digesting enzymes which have specificity as to where exactly they can cleave the peptide bond. Okay, so you are now going to subject this to treatment. You are going to subject this fusion protein to cyanogen bromide cleavage, as a result of which you now get this chain A separately and you get chain B separately, which is removed from its beta gal part. Okay, and eventually now both of these chains are brought together. There are already cysteines here, as a result of which disulfide bond can be formed, and now you get the active insulin. Okay, so this is the way, like you see here, is that you are able to make now a recombinant insulin. So this should just give you an idea as to how, the idea here is to understand that how molecular biology tools can be used in order to make these recombinant therapeutic proteins, because they are proteins, they are coded for by genes. Those genes can be cloned and then they can be put into an expression platform. In this case, the example I have shown you is that of E. coli. So now let us move to the next slide, where I'm trying to show you, for example, how another therapeutic protein, which is called as human serum albumin, okay, can be produced in a different host. Although it is a human protein, as the name suggests, it is human serum albumin. So albumin makes up about 60% of the protein in the human blood, particularly in the serum part, is really the albumin. Okay, and it is a therapeutic protein. It is used for several applications. So let me describe the strategy which can be used in order to clone this. And once you have created a recombinant host, the second stage is to be able to overproduce that recombinant that expression platform which you are going to use in a large, you have to cultivate it in a bioreactor so that large quantities of that protein can be obtained. Okay, now, so in this particular case, what we see here is, again, it is a recombinant plasmid, what you see here, and this blue arc which you see is the single copy of the gene which is going to code for human serum albumin. Okay, so this is a part of the activity which is also being carried out in my own laboratory. So in this particular recombinant vector, there is a single copy of the gene which is coding for human serum albumin, and this particular construct is now electroporated into a yeast system which is called as a Pichia pastoris system. In the previous case with insulin, you had seen that the recombinant vectors were being transformed into E. coli, into a bacterium which is used as an expression host. In this particular case, we are using a yeast system which is a Pichia pastoris. So before I come to this particular part, let me just finish this part of the cycle. So we had first a single copy clone here, and what you see in this particular picture is again a recombinant vector which is constructed. So the maroon part which you see here is the vector backbone, except that what we have now in this particular vector are 3 copies of the gene. All right, so one copy is shown by one arc. The second copy of this human serum albumin gene cassette is shown here, and the third copy is shown here. All right. Now the rationale behind that is that what we have also learned is that higher the copy number is going to be, higher the number of copies of that particular gene which are going to be introduced into the host, chances are that you will also make higher quantities of the protein. Okay, so if you are going to put 3 copies of the gene now, we believe that that host should be capable of producing 3 times as much of human serum albumin than if you only had a vector which contained a single copy of the human serum albumin. Okay, so that was the rationale, and in this particular case, it also worked very successfully in our laboratory. So we have a plasmid, a recombinant vector which is constructed in which there are 3 copies of these cassettes. Okay, now why do I choose the word cassette and not just a gene is because every gene which you are going to put into a vector molecule must be preceded by a promoter, or there should also be a leader sequence in front of it, so that this entire unit, okay, is something which is capable of functioning. Any gene, if it needs to be transcribed first into mRNA, must also have a promoter sequence. So in these constructs, there are the necessary promoters, there are necessary leader sequences, etc., which will allow the protein which is synthesized from here to be excreted into the culture filtrate. Okay, because in this picia, one of the advantages is that it is a yeast system and it has the capability to secrete proteins extracellularly. So that is an advantage, and this is what we make use of. Okay, so we have this 3-copy recombinant plasmid which goes inside this yeast. Okay, now you will recognize that this yeast looks very much like the baker's yeast, and that is why it is called as a budding yeast also. That is Saccharomyces cerevisiae. Now picia also is a budding yeast, so you see a little bud which is coming out, and then you have the rest of the yeast here. All right, so this particular recombinant vector, I use the term electroporation here, not just transformation. And that is when you carry out introduction of these foreign recombinant vectors inside the yeast cell by applying some electric current to the yeast cell, then the process is called as electroporation. It doesn't have to be only yeast cell, but it can be any other type of a cell, to which you are going to electrically first give small voltages, and the idea is that you want to disrupt this cell wall, because picia has a very thick cell wall. Okay, and it's not very easy to get this recombinant vectors inside. So therefore, we give it an electric shock which makes this cell wall permeable temporarily, and then these cells are incubated with the recombinant vectors. So the recombinant vector is going to go inside and it goes into the nucleus. The way these vectors are designed, that it is now going to go inside into the nucleus, into the chromosome, and then it is going to integrate into the chromosome. Okay, so there are special sequences which are on these vectors which will allow them to get integrated at a specific location in the chromosome which resides inside the nucleus. Okay, so these are all integrative plasmids. They are not autonomously replicating plasmids. So this way, this will get inside the yeast cell, and then you will see that mRNA is going to be transcribed in the nucleus, which you just imagine there has to be a nucleus, and then the mRNA comes. What you see here is the endoplasmic reticulum membrane right here. So the proteins are now going to be excreted out, all right, and eventually they are going to pass through these secretory pathways into the lumen of the endoplasmic reticulum, into Golgi bodies, and eventually they are going to be secreted out. So what you see is now a recombinant yeast which is capable of extracellularly producing your protein of interest. In this case, it is human serum albumin. All right, so unlike the E. coli system where you saw that the insulin protein was now residing inside and you had to do the inclusion bodies in one of the cases, in the picia, the advantage is that you have now extracellular proteins. And what has made it possible, okay, is the presence of these leader sequences, which are somewhere here on the mRNA. There are leader sequences which are going to be recognized and which allow this to move really into the transport pathways. Okay, so everything happens post-translationally. So the native protein is now synthesized on the ribosome, but since they have these stretch of small about 19 amino acids which are located now at the N terminus of the protein, okay, they are going to guide it to the ribosome, and then eventually they are going to find their way out of the cell into the extracellular medium. Okay, so in this particular, so therefore going back, so what we have done is that you have a genetically engineered yeast cell, which is a Picia pastoris, which is capable of making this recombinant therapeutic, all right, and it is also capable of secreting it into the extracellular medium. All right. Now, one of the very interesting things is two of the things I'm going to discuss with you in this particular slide is number one is that I do make these 3 copies of the expression cassette and I have introduced them into the yeast cell. But eventually in this recombinant yeast, I would also like to know how many copies are finally integrated into the genome. Because sometimes modifications of these recombinant vectors can also occur within the cytoplasm of the yeast. And then it may not have 3 copies, maybe it just had 1 copy finally. All right. So in order to confirm that, we have very, very specialized techniques whereby the genomic DNA can be isolated. All right. So remember that yeast is a typical eukaryote. The genetic material is organized in the form of chromosomes. So the first thing what we do is that we isolate the chromosomal DNA. All right. And then we have specific techniques which is called as the quantitative real-time PCR, and we are able to determine the exact copy number. Okay, and that is what we have done here. So in the genome, by way of very, very specialized techniques, we are capable of determining what is the copy number. So here when I say C1 clone number 52, we had a recombinant clone which only contained 1 copy. This is the kind of signal what we get. Then we have 2 recombinant clones which are shown here, which contained 2 copies of this expression cassette. And you see that the overall number which we found was much higher. Then these are 2 of our clones which contained 3 copies. Okay, so you are able to find the number of copies, and of course in a very similarly in a related experiment you can also determine what are the level of transcripts which are being made. Okay, so we also found that as the copy number increased, all right, like you see in these 2 clones, these are all 3 copy containing expression cassettes, clone number 2 and clone number 33. They made much higher level of transcripts. In fact, in this particular picture, what I'm trying to show you is not the copy number, but rather the level of transcripts. How many messenger RNAs are being made inside the cell? If you have 3 expression cassettes, or if you have 2 expression cassettes, or if you have a single expression cassette. So you see as the copy number is increased, the level of transcripts, the level of mRNAs, which are going to eventually give you the human serum albumin protein, also starts to increase in a linear fashion. Okay, so that's very, very good, because as the copy number is being increased, we are able to get much higher level of transcripts. So both the things, copy number is confirmed, and at the same time the level of transcripts was also measured. All right, and then what is shown right here is that these recombinant yeast cells, for example, can then be cultivated. Here the picture is that I'm going to cultivate them in a small shake flask, 500 ml shake flask, which contains 100 ml of the medium. And what is shown in the picture, okay, is the recombinant human serum albumin which is being made. And the way to find that out is that the culture filtrate, for example, can be collected, which contains the cells, and the medium, it can be centrifuged. So the cells are going to be centrifuged out, and the clear supernatant which you get, and that is where your recombinant protein is going to be. So that particular sample can now be loaded onto an STS-based gel. Okay, so by way the gel is on the gel, we have way of separating out the mixture of proteins, and what you see here as a very dark band is that of human serum albumin. Okay, so this method is production of this human serum albumin at shake flask level. In the center, I'm just showing you that the culture filtrate can now be evaluated for production of that protein by way of carrying out an STS page. Okay, so the dark blue band which you see is that of the recombinant protein, and at the same time this culture filtrate can now also be injected into HPLC for quantitation purpose. That is, here it's more like an analytical method. It doesn't tell you how much quantity you have made, how many gram per liter, how many milligram per liter of the recombinant protein we have made. So we can also inject it into this high performance liquid chromatography system, and we can quantitate it. Okay, so many milligram I'm making per unit cell biomass. so that quantitation can be done. And then eventually from the culture filtrate, we can also purify this recombinant. HSA has been purified by applying protein purification techniques. And then this particular recombinant HSA is now evaluated for what is the charge on it? Is it really bioactive? That is, even if we have purified this protein, the first thing to evaluate is that does it really have the kind of a bioactivity which you desire? Okay, that bioactivity can be checked for human serum albumin by incubating it with certain cell lines like Vero cell lines or some other mammalian cell lines, and they should promote cell proliferation. That is, you should, after a certain amount of time, you should be able to count much larger number of cells if this human serum albumin which you have made through recombinant DNA techniques is biologically active. Okay, so we can carry out and we can measure the bioactivity. Similarly, the net charge can be measured, and particle size determination. Why? Because if you have a native human serum albumin, it is going to have a certain size because it's a globular protein, it has a certain diameter. But if the protein starts to aggregate together, then in a particle size estimator, you will see much bigger sized particles which are coming. Okay, so you will know that the protein which you have synthesized is now aggregating, which is something which you would like to avoid, because you would like to have this recombinant protein as close to the native protein. And the native protein, as a sample of the native protein, it is also available through many pharma companies. So in our case, we got this from a chemist shop. Human serum albumin is sold in small vials, through these chemists. So we just picked up that vial and we compared our recombinant protein with the native protein. Okay, and currently, by the way, the native protein is produced from human serum. Okay, on the shelf we still don't have any recombinant product for HSA, but a lot of us are working on this. At the same time, we also find out the secondary structure. So what do we mean by secondary structure? Is that, a protein, if you just look at the linear sequence of amino acids, that is called as the primary structure. But then the proteins also fold into alpha helices, into beta plated sheet structures, which can be quantitated by certain measurements, which are called as circular dichroism measurements, which gives you an idea of what is the extent of secondary structure, that is alpha helix and beta strands, so on and so forth. Okay, so the idea here is again to make sure that the recombinant protein which you are making should be very similar to the human protein. Okay, so all that has been done, and we have very good results here. Now let me go to the next slide, and I'm going to tell you that in a bioreactor, because your job really, the job of a recombinant while producing recombinant therapeutic doesn't really end here by constructing a clone, but then we need to take it forward into a bioreactor to see that at large scale also we're able to produce this protein. So the slide actually allows you to understand as to what is the advantage of cultivating a recombinant yeast in a bioreactor. So there are several points which are written on this slide. So first is that when you try to have this recombinant yeast cultivate in a bioreactor, what all do you get? You can provide for a much larger volume for cultivation. Now in the normal laboratory, we have very small shake flasks, at maximum a 2 liter or maybe a 5 liter shake flask. But then a bioreactor can go up to 10,000 liters, and in many cases even higher capacity than that. So you have now the ability to cultivate a recombinant yeast at a much larger scale, and the idea is that large volumes of the product can be obtained. Then you also have a much better control of pH, the amount of oxygen or the amount of air which is going to be pumped into the bioreactor is going to be under your control. So cultivation in a bioreactor allows you to better control your system in terms of availability of oxygen or just air. In addition to that, in many cases, there could be a requirement for making available, for example, the feed may have to be constantly added in a system like a fed batch reactor, where you have to constantly be feeding either a carbon source or maybe some nitrogen into the bioreactor. So a bioreactor can allow you to do that. Then you can also monitor the production for a long period of time without removing too much of the sample, because you may want to monitor the course of fermentation, for which a small volume, let us say 10 ml or 20 ml, can be removed from a bioreactor. And then you can see microscopically, you can examine the culture to see that there is no contamination which has occurred. Or you may want to monitor, for example, how much of extracellular protein is now already in this culture broth, so on and so forth. So since you're going to be dealing with very, very large volumes in a bioreactor, even if I remove 20 ml at any given point of time, it's not too much of a volume to be removed, which can sometimes impact. In a shake flask, if you have only 100 ml and you're going to remove 10 ml sample every 24 hours, you're also changing the nutrient dynamics in this shake flask. So you can monitor the production for a long period of time without removing too much of the sample. And for many things, there are probes which are available where online monitoring is possible. Okay, so for example, as the fermentation proceeds, as the growth of the bacterium or a yeast cell proceeds in a bioreactor, you can constantly monitor what is the state of the pH or what is the temperature. You have all the probes which are inserted into a bioreactor, like we had seen in the previous slide, that there is a bioreactor which allows you to do online monitoring of many different parameters. So in this picture, which is right on the top, is shown if we take these recombinant pichia cells which are making human serum albumin and we cultivate them in a large bioreactor, then I'm just going to explain to you, for example, what all is happening and what all can you see in this figure as a function of time. Okay, so on the x-axis here, what we have is the culture age in hours. So at 0 time, for example, you are going to start your bioreactor. You have a certain volume. Let's say you are starting out with 1.3 liter, and now you add 130 ml of your seed culture, which now goes into this bioreactor. Okay, so in black, what you see here, for example, so there is a scale which explains what is the black curve. So it refers to the cell OD. So when I'm starting at a very low cell OD, but as the cells are going to multiply in the bioreactor, you also see that the cell optical density, which is a function of the cell density directly, starts to increase up to this time. Okay, so if we are starting out with very low OD values, like maybe 1 or 1.5 in our bioreactor, at the end of so many hours of fermentation, okay, 168 hours of fermentation, the cell OD has gone to nearly 900 cell OD units. Okay. Similarly, what you see in the gray here, all right, is the dissolved oxygen. So the scale for this gray color is the dissolved oxygen. All right. So what you see here, that initially, because the cell density is very low, the total amount of dissolved oxygen which is in the culture broth in the bioreactor is going to be very high. But as the cell density picks up, okay, the cells, they also start to consume the oxygen which is available right here. And this trend, for example, also tells you how much of carbon is there. As long as now in these Pichia pastoris, they use in this particular part, they are using glycerol as a carbon source. So as long as there is a substrate which is available, the cells are going to consume oxygen, and that is why there is a drop in the dissolved oxygen. Okay, at this particular point of time, the given amount of carbon substrate, which is glycerol, is completely consumed by the yeast cell, all right, as a result of which there is an increase in the dissolved oxygen. Okay, so at this point of time, I know that the glycerol which was added in the initial stages is all consumed, because you see the oxygen here rises up again. That means the cells are not using oxygen anymore because there is no substrate which is available. At this point of time, we start a second phase in a bioreactor, which is called as a fed batch 1, in which glycerol is now added into the medium. Okay, now the green curve which is shown here is actually the feed fed, because in this particular fermentation, all right, which is called as a fed batch fermentation. That is, after a certain given amount of time, there is some liquid which is coming into the bioreactor. All right, and what you see, therefore, this green line refers to the fact that so many ml, like if you see that over a period from 24 to 36 hours, we have gone to approximately 50 ml of liquid has been introduced into the bioreactor. What does that bring? That is bringing at this point of time additional glycerol. Okay, and this phase is called as fed batch 1. So as soon as you begin to supply, again the oxygen levels are going to go low and you see an increase in the cell density. All right, so on and so forth. So here oxygen status is going to be, dissolved oxygen status is represented by this gray line. The black line is representing your cell OD, and what is represented by this pink line here is, if you see here, the pink line represents the total extracellular protein, because as the cells are going to grow in the feed, we have also introduced an inducer which is called as methanol. Okay, because the system is designed in such a way that methanol is going to act as an inducer. So at this particular point of time, which is called as fed batch 2, FB2, we bring in some methanol also as a carbon source, which induces the system. All right, because our human serum albumin is under an inducible promoter, which can be induced by addition of methanol. So as soon as methanol is introduced into the bioreactor, immediately you start to see now that human serum albumin is being made. Okay, so this pink line here represents that there is an increase in the total extracellular protein. All right, now what is important here is that how do we really know whether human serum albumin is being made or not? So the sample which is removed at this particular point of time, all right, is going to be loaded onto a gel, and the results are shown in this particular gel picture here. All right, so the solid black band which you see on this gel is telling us that it is the human serum albumin, which has a certain molecular weight, 66 kilodalton, is being produced in the culture filtrate. How do I know it is 66 kilodalton? Because on the same gel, I'm also going to run a molecular weight standards. So it is a mixture of proteins which contains a 20 kilodalton, a 29 kilodalton, 43 kilodalton, 75 kilodalton protein. So this is also a part of the same gel. All right, so this is our standard, and this particular one we can also compute that this actually corresponds to a 66 kilodalton protein. So we know human serum albumin is being made in our culture filtrate. Okay, now in the bioreactor, as you see here, that you can continuously give a feed like you saw here. More carbon, sometimes nitrogen, or sometimes something else, in the liquid form can be introduced into the bioreactor. In the blue is shown the temperature. So in this case, the temperature is controlled at a certain temperature, like you see here, 28 degrees. Temperature is constantly maintained in this bioreactor. Similarly, the pH can be monitored, because initial pH is, let us say we are starting at a pH of 6. So the red line here which you see here refers to the pH. So the fermentation started out with about 6 pH, all right, but during the course of the fermentation, pH falls low, and then we are maintaining it at a certain pH 5.5. Okay, so it allows you, in other words, cultivation in a bioreactor allows you to not only monitor, to maintain, and also periodically remove small samples for other analyses, like total protein, or a small sample which I have removed from the bioreactor will allow me to estimate how much of human serum albumin I'm actually making. Okay, so that is the advantage of eventually cultivating it. And number two, remember that for any large scale production, whatever recombinant, whether it is E. coli, whether it is a yeast cell, you do have to cultivate them on large scale in a bioreactor. Okay, so in the laboratory we have small fermenters where we can show that yes, up to 10 liter I have checked, it works fine. The industry will come forward, and then if they are satisfied with your result, then they will cultivate it on a large scale to eventually make large quantities of that particular product. So now let's move to the next slide. So in this particular slide, I'm giving you an example of how recombinant DNA can be used for vaccine development. Okay, now the idea is exactly the same, because eventually you are going to produce a protein in a recombinant host, except that this protein is like an antigen, all right, which is coming from the infectious particle like a virus, and then that particular antigen could be mixed with the adjuvants and some other material, and then it is injected into a human body, which is going to make an immune response against that. And then once you have antibodies against that particular antigen, then the next time the virus is going to attack a person, for example, with respect to hepatitis B virus or something, then the human body is already making an antibody against that, so it is able to neutralize the incoming infectious particle, which also makes that particular protein. Okay, so let us see what are the different stages which are involved in vaccine development. So here I have a picture of a virus, and the virus also contains either its DNA or it could contain an RNA. Okay, a virus codes for a certain number of proteins which are going to be coded for by certain segments of genes on the virus. So all this is already known to you, and today with the availability of the genome projects and so many things like for your COVID-19 virus, because today we are at a stage where the entire genome can be sequenced in a very rapid rate. Okay, so the entire gene of the virus can be sequenced, and then you know that there is only one particular gene which is coding for one protein which will suffice as an antigen. Now in this case, since I'm giving you an example of hepatitis B, the viral DNA could be coding for a large number of proteins. Okay, but we have only excised, or we are only working or looking at one particular gene which codes for a unique antigen, one particular protein which is capable of inducing an immune response in a fashion which is enough to allow you to fight back that infection, were you to be infected by the virus. So here is that little gene fragment which codes for an antigen. This is a hepatitis B antigen. All right, so what we have done now is that here is a bacterial cell, and initial cloning is being done in a bacteria. Okay, but eventually the hepatitis B vaccine which is available today is made from Pichia pastoris. But the initial cloning is done in a bacteria. So there is a recombinant, there is a bacterium here which contains a vector, a plasmid DNA, which you are going to use as a vector. Okay, so what you do is that you make E. coli is cultivated on a large scale, the plasmid DNA is prepared, it is digested, and then you create a recombinant vector like you see here. So the blue backbone refers to the plasmid vector. Then we have also inserted a little part of this gene, this entire gene which is coding for this antigen into this. Okay, so now you have a vector right here. So this recombinant vector is introduced now into a yeast cell, because eventually that is the production platform with which we are working. Okay, so what is shown in this particular picture is a yeast cell, all right, and it also contains its own chromosome, its own nucleus, the boundary of which is shown by this green circle here. Now within this nucleus are chromosomes, and within the chromosomes are is the DNA sequence where this particular recombinant vector is going to integrate. Again, this is an example of an integrative vector, not autonomously replicating vector, okay, which can happily keep itself in the cytoplasm. So this particular recombinant vector gets integrated at a very specific location in one of the chromosomes, and now such a recombinant yeast cell can be cultivated in a bioreactor like you see here. So you have large number of cells which are being made, and the way the construct was made was in such a fashion that this particular antigen is going to be secreted out into the culture medium. Okay, so if we just look at we expand it a little bit to show you that this is your recombinant cell. The gene is going to code for this protein, which is the hepatitis B antigen, and in the initial stages it will be intracellular, but eventually it will be secreted out. So in this fermentation broth, in other words, we have this recombinant we have this antigen which is being produced in large quantity. Okay, so now you have extracellular production in a bioreactor. So the reactor broth is going to be cultivated, it is centrifuged, so that the cells are going to settle down, the culture broth is taken, and then you are going to purify this particular antigen. Then this antigen is now mixed with certain adjuvants or whatever, and that is how the vaccines are going to be made, and that is the vaccine which is going to be bottled now, and it will be available with the doctor, it will be available with the chemist, so on and so forth. Okay, so the basic principles are exactly the same, molecular cloning, because vaccine also means that you are actually cloning a particular gene which is going to code for a product, which is a protein product. And that is how recombinant DNA technology can be applied here. So now let's move to the next slide where I'm giving you an example of gene therapy. All right, so by gene therapy, what do we mean is that today recombinant DNA technology has also been applied in the case of gene therapy. So what do we mean by gene therapy? Supposing a person is born with a certain defect. There is a defective gene, as a result of which the protein, which should be a normal protein, which should be coming out of that gene, doesn't really come out because the gene itself is defective. So the person doesn't make that correct kind of a protein, which could have a very important role to play in a human system. Now, to give you an example, this is also given in your textbooks, that adenosine deaminase, okay, is a protein which plays a very important role in the immune system. And if people do not make this particular enzyme, all right, then they also have a defect in their immune system. Now, conventionally, there are two ways of making up for this deficiency. One is to do a bone marrow transplantation, because this particular enzyme is actually made by lymphocytes, which are a part of the human blood system. Okay, and you know the blood cells are constantly generated by the bone marrow. So if you do a bone marrow transplantation, you also take a healthy bone marrow from one person and you transplant it into this sick person. So due to this bone marrow, all right, it also has normal lymphocytes in it, so it is capable of making this adenosine deaminase. That is how you are going to be able to, you know, make this person healthy by doing a bone marrow transplantation. The second way is enzyme replacement. That is, the person can be given injections of adenosine deaminase from time to time, so that the person is able to fight off this or it is able to make up for the deficiency that they have. Okay, but none of these are viable on a long-term basis. Okay, so what has been done now? Before I give you this concrete example, let me show you what the picture is trying to explain. That in the case of gene therapy, either you can have a direct delivery, that is, here is a part of the DNA which codes for that missing gene in the human system. Okay, and this particular gene is now added to a vector like you see here. So this could be several of the vectors or whatever, and then this is directly given to the patient through injection. Okay, so now you have a recombinant vector which makes that particular protein, which is already now injected directly into the bloodstream. So we are expecting that that the recombinant vector contains that gene. The gene will now be transcribed and translated into a functional protein. Or one could have a cell-based delivery where this particular gene which is coding for that is now cloned into a vector. Then it is going to be you can remove patient's own stem cells, okay, which are defective. Then you take these stem cells in a petri dish and you transform them with this recombinant vector, and eventually you have these transformed stem cells which are capable of making that particular enzyme, and then these are going to be directly going back into the bloodstream. Okay, that is another possibility. So the example which I am giving you here is something very similar to the cell-based delivery. So in this particular case, what you see is that now there is this adenosine deaminase, cDNA. So it is the cDNA which is derived from mRNA, messenger RNA, and reverse transcribed into a cDNA, which is coding for this. It is cloned into a retroviral vector. All right, so you now create a recombinant vector. So this recombinant vector is now transformed, or it is transfected. It is introduced into lymphocytes, which are the sites of producing this adenosine deaminase. Okay, then these lymphocytes are directly injected into a person. So this is something which is called as a cell-based delivery. All right. However, except that, these lymphocytes, remember that they don't have, they can last for a few months, because the entire blood keeps on getting renovated from the human bone marrow. So after some time, these lymphocytes are also going to die out. So at that time, there is a fresh injection which is required of these transformed lymphocytes so that the deficiency can be met. Okay, so in all of these, you will see that the recombinant DNA tools are extensively used in order to make these vectors, lymphocytes, etc., and then they can directly be injected. So this way, the gene therapy is, is possible. Okay, now last point which I just want you to familiarize with, we don't have time to cover all the different steps, but the recombinant DNA or the molecular techniques which are associated with the recombinant DNA technology, such as the real-time PCR, etc., they are also very useful and they can be used in the diagnostics. Like today, when one has to go for a COVID-19 virus, and even if the infection is in early stages, and they have only a few mRNAs which are coming out from the virus, but you have the tests whereby a small sample is removed from your throat, or from the nasal swabs are taken, and from here the nucleic acids are extracted, and then they can be converted into cDNAs. And there are specific primers which are only specific for one of the proteins of the COVID-19, like spike protein, for example. Okay, now that spike protein is also coded for by a gene, so that on the basis of that gene sequence, you can design primers. Okay, so you have a forward primer which corresponds to the 5' terminus of the gene. You have the reverse primer. So these primers can now be used to amplify out. So even if there were few mRNAs which were already in the body, they are transcribing, then when you do this RT-PCR test, the test is going to be positive because you are making use of the PCR based technology to do that. Similarly, for dengue virus also, there are specific primers which are going to be available, which can be used for detection in very early stages. All right, so here we are not making any recombinant therapeutics, but many of the techniques which we have learned in the recombinant DNA, like PCR or like reverse transcription followed by PCR, etc., or real-time PCR. So many of these techniques can now be applied in the area of diagnostics. So finally, I would like to summarize what all we have learned in this lecture is that the tools of recombinant DNA technology have been successfully applied for development of recombinant therapeutics, including monoclonal antibodies, in which they are largely produced through mammalian cells. And vaccines can also be produced, like you saw the hepatitis B vaccine, which is being produced in yeast, Pichia pastoris. Then recombinant DNA can also be applied for gene therapy, like you saw, or it can be applied in diagnostics. It can also be applied certainly for transgenics, okay? Because here again, if you want to create a transgenic cattle or transgenic sheep, then the route is exactly the same, that the specific genes which are going to give a unique function finally are going to be cloned into an animal virus. It is then introduced into an animal body. So that way, you do bring in some other kind of characteristics which are not quite natural to that particular animal. So this way, transgenic animals can also be created. So with this, we end this lecture on applications of recombinant DNA in medicine.